let's look into the physiological framework of cell deterioration in animals as they mature and age. Imagine a world where we could slow down the relentless march of time, preserving the vitality and function of our furry friends' lives well into their golden years. Though the fountain of youth may be elusive, recent breakthroughs in our understanding of the biology of aging offer promising paths towards this goal. Today, we will delve into the physiological underpinnings of cellular deterioration, uncovering the key mechanisms that drive this process and revealing potential targets for interventions to promote healthier, longer lives in our animals. First, some key elements of aging. As we get older, the individual cells and tissues that make up our bodies start to break down and lose their normal functions. This progressive decline happens gradually over the course of our lifetimes. Second, along with the deterioration of individual cells, the overall systems and processes that keep our bodies functioning properly also become impaired with age. Our organs, metabolism, and other physiological functions don't work as efficiently or effectively as they once did. Third, due to breakdowns happening at the cellular and physiological levels, our bodies become more vulnerable to developing various diseases and health problems as we get older. Conditions like cancer, heart disease, neurodegeneration, and organ failure become much more common in our later years. The physiological mechanisms of aging include stem cell exhaustion and altered intercellular communication, which means as we get older, the stem cells in our bodies, or the master cells, can develop into different cell types, start to wear out, and lose their ability to regenerate and repair our tissues. At the same time, the communication between our cells becomes disrupted. These changes make it harder for our bodies to renew and maintain healthy tissues, leading to the signs of aging. Second, oxidative stress and mitochondrial dysfunction. Our cells have tiny structures called mitochondria that act like a powerhouse, generating the energy our cells need to function. But as we age, these mitochondria start to malfunction and produce harmful compounds called free radicals. This oxidative stress damages our cells and their genetic material, contributing to aging and age-related diseases. Lastly, telomere attrition and genomic instability. At the tips of our chromosomes are protective caps called telomeres. Each time a cell divides, these telomeres get a little shorter. Eventually, the telomeres become too short, causing our chromosomes to become unstable and leading to DNA damage throughout the cell. This genomic instability is another key driver of the aging process.
Hello, my name is Shakoya Hoyt, and I will be going over the slide about stem cell synthesis and altered intercellular communication. So you ask, what are stem cells? Stem cells are to regenerate and repair tissues that may have been damaged. Stem cells can also be directed to differentiate into particular human usable cells throughout the body. The body can regenerate those cells and restore tissues that may have been damaged or impacted by disease through the process of synthesis. Those who may have had leukemia, Hodgkin's disease, and other tumorous diseases may have been able to benefit from stem cell therapy. But as we age, adult stem cells lose their function, potential, and become less healthy. Some examples of stem cells in our body include self-renewing cells called somatic cells that are found in human tissues and organs during development. Neural cells are multipotent self-renewing cells that are mostly presented in the central nervous system's neural tissue. Mesenchymal stem cells are also multipotent stem cells with the capacity to differentiate into several subtypes of tissues, which includes osteoblasts or bone cells, cartilage cells, myocytes or muscle cells, and adipocytes or fat cells. With great capacity for regeneration, adult skeletal muscle stem cells, also known as satellite cells, can arise following an injury or a consequence of memory B cells and naive T cells. Hematopoietic stem cells are those that produce blood through the process of poiesis. They are located in red bone marrow, which is the marrow cavity of most bones. Hematopoietic stem cells also make immune cells for organisms. Altercations can be caused by external factors and reduce their capacity to renew. Stem cells can be altered or reversed by systematic or circulatory components obtained from young blood via anatomical joining or parabiosis. One treatment, referred to as retrotransposon, requires mobile DNA segments that have been associated with aging-related cellular dysfunctions and have the potential to cause genetic inability. They do this by converting RNA into DNA through reverse transcription. Furthermore, parabiosis allows for modulating the molecular structure of the tissue's environment rather than inducing the cell alone. As I mentioned before, systematic or circulatory components derived from young blood by anatomical joining can be employed to change or reverse. It's interesting to also note that parabiosis has been shown to rejuvenate the liver tissues, muscles, and the brain in elderly animals. Other methods include cellular reprogramming, or iPSCs, which is a kind of potent stem cell that can be produced from adult cells directly and allow the biological clock to be edited and reset by eliminating traits that come from disease cells associated with aging. And that concludes my slide. We will now be moving on to the next one.
Let's take a look at the physiological effect of oxidative stress and mitochondrial dysfunction. Here, mutations accumulate in the mitochondrial DNA over time, impairing the mitochondria's ability to produce energy efficiently. The mitochondrial dysfunction also feeds back to generate even more harmful reactive oxygen species, or ROS, creating this vicious cycle. The excessive ROS generated overwhelms the cell's antioxidant defenses, leading to oxidative stress. This oxidative damage to cellular components contributes to the functional decline that we see during aging. Some potential interventions are seen in long-lived animal species that tend to have higher basal activities of antioxidant enzymes. So one possible intervention would be supplementation with antioxidants like vitamin E, vitamin C, and CoQ10 to help neutralize excess ROS. Another one would be to restrict calories. Calorie-restricted animals show reduced mitochondrial ROS production and lower oxidative stress to mitochondrial DNA. The mechanisms likely involve metabolic adaptations that decrease electron transport chain activity and increase mitochondrial biogenesis and turnover.
I want to talk about a significant proponent of aging, which is genomic instability. As we age, our cells pick up the genetic equivalent of scars from internal and external threats. This damage presents itself in a multitude of ways, from mutations to chromosomal alterations to telomere degradation. While our bodies have mechanisms to repair this damage, these mechanisms decline as we age. One of the key players in this process is the telomere, which functions as a protective cap at the end of our chromosomes. Each time a cell divides, the telomeres shorten, and when they become critically short, cells can no longer divide and enter what is known as cellular senescence. This plays a significant role in the aging process. While the enzyme telomerase can preserve telomeres, it is absent from most somatic cells. Reducing the sheltering complex, which guards telomeres from being recognized as double-strand DNA breaks and thus halts chromosome fusions, illustrates how essential it is to preserve telomeres and chromosome fitness. The systemic viral transductions of telomerase into mice has been shown to delay normal physiologically associated aging and thus offers an increase in lifespan. These findings raise hopes for potential new therapeutics to prolong a healthy lifespan by manipulating telomere maintenance pathways.
As animals age, their cells age with them, meaning that their bodily functions are soon going to stop working how they should. As they get older, their cells start to degenerate, and they will soon become susceptible to any disease that their body seems to contract. Looking at this image, it shows a young mice's cell versus an old mice's cell. Their shape and everything around them seems out of place. You all, as future scientists, should continue your research and gain more knowledge on more ways we could possibly prevent older animals from dying of degenerative cells. We love animals, and more so our pets, and seeing that as they age, they become more of a target to diseases. We want to make sure that our pets are active and up to date on all vaccines, just like us. We want to live a long, healthy life, so why can't we find more ways to do the same for animals?